After the roots, now let us discuss about the stems. Firstly, we are going to take the stem of a dicotyledonous plant. For example, you have a sunflower plant, you're going to make its transverse section, you're going to observe it under microscope, and a part of that stem would look like this. Now, what this lesson demands from you is to pay attention to the part of that stem, what all parts comprise that stem. Secondly, you need to have an idea of the diagrammatic representation. That means the syllabus asks from you to diagrammatically practice this particular presentation of cells. So, you have to practice the diagrams and learn what is located where. So, for that you need to know what is present where. So, we have 5 components of the dicot stem that are listed over here. One by one we are going to understand the importance of that particular component and where it is located, because we are dealing with anatomical references. So, we are going to see if a dicot stem, its transverse section is taken, of course it is a stem, because it is cylindrical, what you would observe under a microscope would be a circular arrangement, something like this. Now, as you can recall from the first lesson where you had a projector presentation of these various slides that you make of various anatomical regions of a plant, you saw that the dicot stem would be something like this, and there was a wavy pattern that, not like this, I would use another concentric circle because you have to be aware of how to represent it diagrammatically. This concentric circle would be used to give a wavy pattern that is somewhat stellar arrangement, you can say. And stellar arrangement, because it is not completely descriptive, I would suggest you to keep in mind that there is a wavy arrangement in such a way that it looks like some sort of wave that is transcending inside the stem. Now, this wavy pattern, this line that I have represented, this wavy pattern is because of a wavy endodermis. Okay, now this is the stem that you would observe under the microscope. This part, that black line inside which is wavy, this is endodermis, and as you can very well associate now and understand that the outermost would be epidermis. So, we have an epidermis and an endodermis. Epidermis is circular while endodermis is wavy. In between the epidermis and endodermis, we have a region of parenchymatous cells which is known as cortex. So, the first 3 things that we have to remember is that the outermost layer is epidermis, which is followed by parenchymatous cells of cortex, and the cortex ends in a wavy endodermis. Now, what lies below this wavy endodermis? Wherever there is a trough in the wave, you know what is trough and what is crest. So, where there is a trough in the wave, that region would house a vascular bundle. Now, what sort of vascular bundle, we are going to discuss that, but for now you can just remember that a vascular bundle would be located over here, one would be here, one would be here, like this. You are getting a pattern. Alright, this is the roughest representation just in order to have an idea. So, these are the vascular bundles. What we have done till now, epidermis followed by cortex followed by endodermis, in between we have a vascular bundle, and in the center we have a large conspicuous pith. So, this is what a dicot stem is basically, an overview if I would say. Now, what we have represented over here, it does not seem so simple as we have drawn here. So, we take a part of this region and draw it over here. This is what has been done. This is the entire stem. A part of the stem is being magnified and observed under the microscope in order to understand the arrangement. Because you can see that this region is a repetition of this, then followed by this, this would be a repetition, this would be a repetition. So, it's better we consider this much region where we are having all the components represented in an array, and we can understand that. So, taking this part under the microscope, we observe the arrangement in such a way where we have, now I am going to rub it, I hope it is clear. This was clear even in the first projector lesson where you got an idea what a dicot stem would look like under the microscope. Now the outermost layer, as you can see, this red line that lines the outermost layer, that is single layered cells. So the single layered cells would be called as epidermis, outermost layer, which will have a layer, thin layer of cuticle that would surround it. Now this cuticle is there, epidermis is there. From the epidermis you can see that there are multicellular projections, unlike epiblema where we had epidermal cells only elongating their cell walls and forming the root hair. Over here we have multicellular projections known as trichomes. Please remember this term. These trichomes are otherwise known as root hair, stem hair. Now, why is this name stem hair, though it is not having any parallel with the root hair? Just because we use the term root hairs over there, and because they are epidermal extensions, these are also epidermal extensions, so we call them stem hair, alright. But the point of difference is that they are they are multicellular firstly, secondly they are covered with cuticle, and both the conditions are not present in the case of root hairs, which is unicellular, and at the same time there is no cuticle which is present. So the first point of difference you have over here, that there are trichomes which are multicellular. Do remember that I am mentioning over here. So as long as you see me, the multicellular word is also in your sight so that you remember it. So the trichomes are multicellular stem hair extensions of the stem epidermis. Moving further, we have the cortex. Now in the cortex, it's better if you understand it in terms of three regions. See, the innermost region is the endodermis. Over here, the cortex ends. The other name for endodermis is starch sheath. Remember it. Now sheath is something, single layered sheet. This starch sheath, why endodermis is called starch sheath? The reason is because its cells are rich in starch. So we have the endodermis, which is otherwise known as starch sheath. Talking about this region, from the first layer below the epidermis till the endodermis, we have the region known as cortex. So this, not over here, over here I am writing that this region, this green region is known as cortex till the endodermis, okay. Now this cortex has the uppermost layer, 1 or 2 or 3 layers I would say, known as hypodermis. See, all this is cortex only, but we have the different terms. This is hypodermis. From anatomical point of view, this hypodermis is made up of the cells which are collenchymatous. Now try to recall what collenchyma was. Collenchyma was a type of plant tissue, simple permanent plant tissue, which had cells in such an arrangement that they had wall thickenings, angular wall thickenings. They were living cells. They had angular wall thickenings where they had deposition of hemicellulose and cellulose. So these regions having hemicellulose deposition, wall thickenings where the cells are joining, that part forms the hypodermis in the dicot stem. Alright, now this is what is my way of representing collenchymatous thickenings. As you can see, you can just put these as angular thickenings. Now because there are angular thickenings, the hypodermis would be responsible for providing extra strength to the plant. Okay, one point of difference that we have from the root, that in the case of a stem that we are studying, is that there are no thickenings, no such arrangement in the case of cortex. And because we are studying the stem and because it is dicot stem, the main points of difference would be at the level of dicot stem and monocot stem. So do remember, the hypodermis in the case of dicot stem is collenchymatous, where the cells are having parenchyma. That's why I have used green color for the representation. But at the same time, they have angular thickenings and they are responsible for providing the mechanical strength. Now, this is hypodermis. This hypodermis varies in the case of monocot stems. How, that we will come to know. But for now, do remember it is made up of monocot. It is made up of collenchymatous cells. Moving further, there is a layer. There are many layers, in fact, of parenchymatous cells which may have oil ducts in their arrangement, which may have many intercellular spaces or less intercellular spaces, and that particular region forms the cortex. So, we have hypodermis, cortical layers and endodermis which form the cortex basically. Alright. Now, in the cortical layers, you have to remember that the cells would be parenchymatous and they may have oil ducts which are responsible for releasing oil, and at the same time they are responsible for performing photosynthesis as well. Last layer is the endodermis, which is also known as starch sheath, and the starch sheath is rich in starch, the cells which form, and do remember the cells are barrel shaped, tightly packed, and this forms the endodermis. Now, till here we are done. Talking about the third component, that is medullary rays. See, this was parenchymatous, this was collenchymatous and parenchymatous. These medullary rays are going to be exclusively parenchymatous. And one point you have to remember, firstly, where they are located, that you have to remember, of course. And secondly, you have to remember what would be the arrangement of cells and what type of cells would be present. Now, these larger cells larger than the cortical cells, which are arranged between two vascular bundles. This is one entire vascular bundle. See, this part is a vascular bundle, this part, okay. From here till here, I am representing it. This would be a vascular bundle. Below the endodermis, this was the wavy pattern that we discussed in the beginning. The cells which line the vascular bundles, the cells present between the two vascular bundles of that wavy pattern, they form the medullary rays. And you have to remember one thing, that these medullary ray cells are extraordinarily large than compared to those of the cortex. Now, coming to the main part, that is vascular bundles. We have in vascular bundles, we remember from our previous knowledge that in the vascular bundles of a dicot stem, first of all, the bundles are conjoint. Vascular bundles are conjoint. They have an open type of arrangement. And why do we call a vascular bundle open? If it is having a cambium in between. Okay, such is the arrangement that phloem lies towards periphery, that is outside, and xylem lies towards inside. There is a cambium which is present, there is a phloem, there is a xylem. This phloem is surrounded by a patch, not surrounded, or you can say there is a cap-like patch of pericycle as well. Okay. Next, vascular bundle would be such. It will be having cambium, it would be having phloem, it will be having xylem. There would be a patch of pericycle again. And what lies in between these two is known as medullary ray. Okay. Now, these medullary rays are parenchymatous, and now what we are going to study is a vascular bundle. Now, let us see how this has been represented in the diagram. First of all, we have inner to the endodermis, we have this arrangement, this red part that is known as pericycle. Now, pericycle lies just beneath the endodermis, and you have to remember one thing. Now, supposedly pericycle is this cell, single cell, that is a parenchymatous. It will have sclerenchymatous C patch, something semi-lunar, you can say, patch of sclerenchyma. Okay. Now, when we talk about sclerenchyma is responsible for providing tough base to the cell. So, these are pericycle cells. Moving further, these parts, that is beneath the pericycle, we have phloem. Now, phloem is the complex tissue that has 4 types of cells. I need not to make you recall, still we can remember them. Companion cells, sieve tube cells, phloem parenchyma and phloem fibers. So, the region which is responsible for conducting the food material within the plant stem lies below the pericycle, and that is known as phloem. Then we have tube representations, that is xylem. As you can see, these red tubes that are arranged on one another, they are known as xylem. This is again a complex plant tissue which is having the arrangement in such a way that the metaxylem lies towards the periphery and the protoxylem lies towards the pith. Now this is metaxylem because it is having larger diameter, as you can see. Then we have the protoxylems. So in this case, you have to remember that what would be the arrangement. If you can recall, we said that the reference is made in terms of protoxylem. Now because the protoxylem lies towards the pith, so the arrangement is end-arc. End-arc arrangement of metaxylem and protoxylem is observed. This is the xylem basically, which is arranged in an array. This is the phloem. Now in between lies 2-3 layers of vascular cambium. Now this vascular cambium has an important role to play when the plant has to show secondary growth. Which plant? Dicot plant, because only dicot plant shows secondary growth. Of course, there are few exceptions in the case of monocot plants, like the palm trees that we have in our houses. They are having secondary growth. Keeping aside the exceptions, you are going to find this 2 to 3 layered rectangular cell cambium, vascular cambium, present between the phloem. This part is phloem, alright, between the phloem and the xylem. Now this phloem and xylem region, they are basically the conductive tissue. In the conducting tissue, we have the vascular cambium that is responsible for secondary growth. That means it would give rise to phloem and xylem, but it would not be rising from the primary meristem. And inside to all this lies a huge conspicuous pith which is having lots and lots of intercellular spaces, huge area, and there is arrangement of all these vascular bundles in such a pattern that it looks a wavy structure where all these are arranged in a concentric way. Now the vascular bundles are conjoined, you have to remember. The arrangement of xylem is end-arc because protoxylem lies inside. There is a vascular cambium present in between, that means the vascular bundles are open. Then we have a pericycle which is present just above the phloem, followed by the endodermis, which is also known as starch sheath, starch rich cells which are barrel shaped and closely placed. Then we have the cortical layers which have the parenchymatous cells and oil ducts present in them, followed by the hypodermis. Hypodermis which would be rich in collenchymatous cells which have extracellular thickenings, and then we have an epidermis covered by a cuticle. The epidermis might have trichomes, thin stem here that you might have found if you have a keen interest in reading plants. So this is all about our dicot stem. You have to keep in mind what distinctions you can put while representing the diagram. Once you're clear what is present where, it would be easy for you for getting a better diagram. So this is all you have to understand when we study the transverse section of a dicot stem.